A few more questions to do on chapter 14, so we need to talk about bowelism and we need to talk about Parkinson's and these kinds of questions. And then we are going to launch into the next chapter, which just went up last night. Paradoxically, I am sleep deprived today because I was working on this until late last night, partly because I got distracted by some other things related to the course. But in any case, that will be a sort of our focus for the coming week, is this chapter on brain rhythms and sleep. And I think it's a really good chapter. As far as future things, I would like to do a quiz on chapter 14. So I'm scheduling that for Friday. If there is the usual slew of people that are not going to be here Friday, please let me know and we'll make alternative arrangements for you. You guys? So we'll figure that out. When do you guys leave? Thursday morning? So, okay. Well, let's talk afterwards if you can. And then let's see, and then the homework for this chapter is not due until next Monday, which is the 10th. And then other things, partly what I was doing last night while I was not working on your slides and I was supposed to be, is I was looking ahead at these other chapters, and I have to be honest, I felt kind of like a bit let down about the emotions chapter. I'm not loving it. So we may, but on the other hand, I really like the mental disorder ones. I think it's good. It's a lot of mechanism. It's got a lot of meat to it. Whereas the emotion one seems kind of like, I don't know, it's less neuroscience-y and seems more psychology, which is fine, but it's not really something I feel comfortable doing much on. So I could basically say, hey, there's the limbic system, and there's one really cool study in cats. But aside from that, I'm not sure how much of that we're going to end up doing. But that will also mean that we can spend more time on that last chapter and give it a good thorough treatment. So I'm sorry, that side of the room that all voted for emotion, I'm not sure how much we're going to do. But I think what we will do will be interesting and worthwhile. And really a very nice way to wrap up the whole semester. So I'm actually really plussed about the mental disorder chapter. So any questions about anything before we launch into the homework questions? Right, seeing none, let's move on. So we were talking about, we did question 1, 2, 3. So now we're talking about question 4. So question 4 reads, there are two diseases involving motor ganglia. Two of the diseases involving motor ganglia are Parkinson's, which is loss of the substantia nigra, and bowelism, which is loss of the subthalamic nuclei, both of which we kind of skipped the subthalamic nucleus when I was talking about in lecture, but both of these are present on that diagram. So what are symptoms? Why is it that loss of that brain area would lead to those disorders? So does anybody remember where we left off last time? I cannot remember for the life of me what group we were on. I kind of feel like maybe it was you guys, but I don't know. Do you think it was you guys? Who would like to answer this one? Let's put it that way. That was like 4 days ago, and that's a long time ago. So I don't remember. This is a great one. You guys are all clinical people. This is like clinical stuff, kind of. Yes, thank you. Did you say matic or automatic or autonomic? Matic fits better. Okay. Yeah. That's right. I mean, bowelism refers to like ballistic movement. So that's kind of like, these kinds of things where there's these sort of ballistic flights, kind of like a ballistic IBCM missile, but instead it's your arm behaving ballistically. So it is these sort of flailing, undesired movements that can be very frustrating. I mean, just incredibly frustrating for the person that has them. So why is it that loss of this specific brain area cited above results in these symptoms? So what did you get for, like, this is the hard part of it, but. Do you have a proposal for why loss of these would result in the disorders, though? That's the tricky. Yeah. Before we get to the dancing one, let me just pull this up. So I think this is really interesting, and this is why I like this question a lot, because I think you can actually, to some degree, make out at least some of the symptoms for the two disorders. So for Parkinson's, the main thing that happens in Parkinson's is you have loss of all these dopaminergic neurons that are found down here in the substantia nigra. So if these guys go, then you no longer get them here. That is that neuron that normally sends axons, among other places, up to the putamen here where it releases dopamine. And that, as the plus sign indicates there, that's an excitatory synapse. So usually the role of the substantia nigra is to increase the excitation of the neurons up here in the putamen. So if you lose the substantia nigra, you're losing that excitation, which means it's harder to get this black neuron excited. The only source of excitation at this point really would be from these frontal cortex neurons, and usually there's sort of multiple sources of excitation on these guys. Now you've got sort of half the excitation, if you will. So if you lose that excitation, that means it's hard to get this guy going, which means it's harder to send signals down the line through this motor loop. I mean, normally what happens is this guy inhibits this one, the globus pallidus neurons, and then the globus pallidus neurons are now unable to inhibit the ventrolateral nucleus of the thalamus, and therefore you're disinhibiting this and allowing the signal to go. But like the key thing here is that if you shut down this excitation, you can't send signals through this pathway anymore, right? Or at least it's more difficult to. And the function of this pathway, if you remember, this is to create movement. So if you're going to initiate a new movement, the idea here is that you're sending a signal through this motor loop pathway. So if you impair that signaling by losing your substantia nigra, it's harder to start a movement now. And that is one of the major symptoms of Parkinson's, is this sort of rigidity or inability to initiate motion. Like once they get going, they can kind of keep going sometimes, but it's very difficult to start motion. It doesn't overtly explain the resting tremor, and that's frustrating to me, but I don't have a good explanation for the resting tremor. I'm sure there are some out there, I just don't get it. But at least in terms of that common symptom of having a hard time starting a movement when you have Parkinson's, this explains that pretty well. Subthalamic nucleus embolism, I think this also follows nicely. So the idea here is, here's your subthalamus. So if you're losing these neurons, these guys normally send excitation to the globus pallidus neuron here, which then in turn normally will inhibit the VL nucleus and prevent sort of unwanted movements. So if you lose these neurons, you no longer excite the globus pallidus neurons, which means you're no longer inhibiting these neurons up here in the thalamus, the ventrolateral nucleus of the thalamus. So these guys are disinhibited unintentionally, sort of, and as a result you get these unwanted sort of ridiculous feeling, I'm sure they must feel ridiculous, motions that occur, these ballistic motions that occur under this pathology. Does that make sense? Hopefully it does. I mean, questions on how those things work? Because I do think you should be able to reproduce that. Like, you should understand how loss of substantia nigra results in harder to start a motion. You should understand how loss of subthalamus results in disinhibition of the ventrolateral nucleus to cause unintentional motions. Okay, I'm getting some nods. So I think it's neat, because I think you can directly see why those symptoms, at least of those conditions, would result from the loss of the specific neurons that we were talking about. How about this last one? So what'd you put for this one? This one's interesting. Send. This is kind of the argument. I mean, it's not. There's not a. This is why I said please try to explain specifically what's happening. And there's not like a really obvious, completely accepted explanation here. But there does seem to be a trend. This is sort of an ongoing trend in Parkinson's patients, is to give them these opportunities to move well, and they're doing it because they are in this situation where they're already starting to move. And so if they can get sort of those initial movements, it seems to facilitate future movements. And again, like we talked about, the big thing that's hard to do with Parkinson's is initiate a movement. So if you already have movements occurring, then perhaps it's easier to send signals through the motor loop, or maybe there are ways to bypass the motor loop once you've already activated the actions. It's kind of that's the explanation I would try to put forth for this thing. I'm not saying I completely understand it, but that does seem to be that seems to be an explanation that's accepted for why this might work. It's pretty amazing. I mean, you kind of run into these situations where these people are, all but unable to move. They're just sort of sitting in a chair all the time. They can barely move even in the chair. And then, you start them in these sort of dance therapies, and some of these patients at least are able to start at least moving on the chair, if not actually getting up and moving around the room. And it's just, I mean, it's kind of incredible. Okay, number 5. So let's see. So it asks you to read this box describing these different disorders, and basically what I'm asking you to do is compare them and try to explain clinically how would you distinguish each of these from the other. So I didn't include bowelism because to me it's a little bit like Huntington's in terms of symptoms, but included these other 3 that were listed in that box. Anybody feel like tackling this one? I can call in a group if not. Yeah, okay, go ahead.
I have for Huntington's repetitive movement, more like big movement, like bending an arm. And it's involuntary.
Yeah, what about cerebellar intention tremor?
I have fine movement in distal limbs when moving from one point to another. And then Parkinson's pushing and shaking while this.
Yeah, I thought the Parkinson's video is kind of interesting, right? Because it really demonstrates, I think, how much of it is a resting tremor. So, the person sitting there, and if they're trying to basically rest, they're just, twitching around everywhere. But if you ask them to move their arms up, at least in this stage, this person is still able to initiate movements. They just kind of lock solid until they're resting for a while, and then it starts to twitch. But I mean, again, it's the resting time when you tend to have most of your twitching going on. As opposed to some of the other ones where you have it more sort of during the action itself. Have I put anything different for any of these other ones, or is that more or less what you had? Why do you think cerebellar intention tremor is happening? So like in the video, it's, this guy where one side, he's fine. He can, touch his nose back and forth, no problem. The other one, it's just, why do you think that's happening? Any ideas? Huh? Where though? Like what part of his brain do you think is damaged, and what? I mean, yeah, probably cerebellum. So like why would damage the cerebellum cause that symptom, that sort of inability to do fine precise motor behavior? It can be anybody, but what do you think? Nobody wants to say. So what's the cerebellum do? Yes, sir. What do you think?
Relays primary messages between cortex.
Well, the pons is relaying that, right, between cortex and cerebellum. But like, what is the cerebellum's function in getting information from the cortex? And like, what's it trying to do? Yeah. Yeah, so it's getting motor information, which tells it what you want to do. And it's getting sensory information that tells you what is actually happening. And it's comparing them and sending corrections. So if you have damage to your cerebellum, which is what this poor guy had, then you have a situation where it's very difficult to coordinate those kinds of fine particular movements because you're sending motor impulses, but you don't have this mechanism in place that efficiently compares the motor actions you're trying to do and what actually is happening and sends corrections. That's what the cerebellum does. So if your cerebellum's broken in this case, then you have that kind of intention tremor where it's very difficult to actually perform a task. And that's sort of in opposition to the Parkinson's tremor, which is part of why I included it here, where the Parkinson's tremor is more of a resting tremor. But when you're doing intentional movements, the Parkinson's patients can actually do the pretty precise movements because their cerebellum's fine. The issue is initiating the movements and then that resting tremor business, which is very frustrating. So again, the idea is, I think the cerebellar intention tremor does a really nice job of showing exactly why it is, or exactly what the function is of the cerebellum, at least in terms of motor actions. Let's see. And then Huntington's, we also, yeah, I think that's pretty much the case in Huntington's. At least symptoms-wise, it's very similar from the general bolisms that we were talking about in the last question. It's a tough video to watch, that one. Okay, number 6. I love this one. So this is this story about this guy. Came on the news a couple years ago who, he doesn't have a cerebellum. And you can see that in the pictures, which is great. I just think those are marvelous. So here's you have MRIs of of this patient versus normal brain, and oh my God, like there's just no cerebellum there. That's incredible. So in any case, symptoms. So like who'd like to tackle this one? You guys really don't want to talk today. Yes, Rachel, go ahead. Yeah, and we were just talking about cerebellum's function in terms of motor function. So that's those kinds of like walking, especially walking, but also talking, standing, those kinds of things. That makes sense as far as missing cerebellum. This other one's kind of cool. Where loss of ability to interpret social situations. Like what? What's going on with that? So that kind of segues to the next question, which is why is it that the story of the cerebellum in chapter 14 is incomplete? So what'd you put? I'm going to put. And what'd you put for this last one as far as this? I love this because it wasn't even in the story, and I'm staring at this thing. I'm like, what the hell? So what else is this guy? What's weird about this person's brainstem? Did you see that? Yeah. Why would that be? Mm-hmm. Yeah, so we talked about in the chapter how a major function, in this chapter, how most of the pathways that travel from the cortex into the cerebellum, well, I should say it over here because there's cerebellum. Most of the things that are going from the cortex of the cerebrum into the cerebellum are relayed through the pons. And so what this is indicating is how large a chunk of the volume of the pons is dedicated to little nuclei that are receiving those centers and then relaying those signals up to the cerebellum. So in this poor guy, he doesn't have a cerebellum anymore. There's this big empty void. But also his pons is just like this shriveled mass of what it normally is. It's really not distinctively different from the medulla anymore. Whereas in a normal person, the pons is much, much thicker and hardier because of all those nuclei that are in charge of relaying up to the cerebellum. So I think that's pretty cool. You can actually see that function there. Did anybody put anything different or additional for A and B you would want to throw out? I mean, definitely the social system or social situations things seem to be a challenge. I remember that, like, the picture even in there showed something like less talking, more working. So, like, there were sort of consistent problems with focus, ability to manage more than one thing at once, including like listening at the same time you're talking, these kinds of things. Clearly, the cerebellum is involved in a lot of functions that go beyond just motor pathways. And all your book really, in that chapter anyway, describes are these motor-related functions of the cerebellum, but it's involved in general in lots of sensory processing. A classic example I usually talk about in my anatomy course is if you're listening to words that sound similar to one another. So rapid and rabid and rabbit, all three of those words, they're pretty similar sounding. They're difficult to discern. Your cerebellum seems to be distinctly involved in them, in those sorts of interpretations, though, because people that have damaged the cerebellum often have difficulty interpreting those sort of fine levels of speech. And as further sort of backup for that, if you look at MRIs or other brain imaging studies that are looking at which areas of the brain are active during particular tasks, most of the time when you have sort of fine sensory distinguishing types of tasks, which it could be visual, it could be auditory, much of the time the cerebellum seems to be getting involved because it's showing increased blood flow during those times. And that's usually a good indication that that part of the brain is operating. So cerebellum does more than just motor function, even though that's really all we're hitting in this chapter. Okay. Any questions or anything about any of these questions we did this chapter? All right. Well, in that case, we're starting to look at kind of whole brain function. So how is it that whole brain operation manifests in particular behaviors or ways in which our central nervous system operates? So this chapter, I think, is a nice introduction to that because it's talking about whole brain functions, and it's talking about things that all of us have really distinct experience with, and that we're going to be talking about brain rhythms and sleep, and to that degree, we're also going to be talking circadian rhythms as part of this discussion. So this is the outline of what we're going to be hitting. So we're going to start with sort of an introduction to brain activity and brain rhythms, and we're going to spend a lot of time talking about EEGs, electroencephalograms, and how we measure them, what they're actually telling us. Then we're going to move on to talking about sleep. So stages of sleep, why we sleep, how we sleep in terms of neurological mechanisms behind sleep. And then we'll segue into talking about circadian rhythms and the specific role your book emphasizes, and we will too, the specific role of the suprachiasmatic nucleus as well as clock genes that are operating within that suprachiasmatic nucleus that seem to dictate the circadian rhythms of our entire body. So how does all that occur? And then there will be homework questions that follow. Get the homework's due next Monday. So the idea here is that every organism pretty much on the planet has some kind of intrinsic rhythm. And I said every organism, I mean certainly all the multicellular organisms and probably a lot of the unicellular ones do too. They have these intrinsic rhythms that cause them to behave in certain ways at different times of day. And certainly we even have circannual rhythms that cause animals to behave in certain ways at different times of year. We're really going to focus on the circadian rhythms. At their most basic level, usually what happens is you have a situation where animals will be more active at a certain time of day that they're sort of optimized to be active in, and they will tend to be less active at other times. So for example, here in this cricket, what these little dark spaces are indicating would be chirps of this cricket, but they're basically recording a cricket. Each line is a different day, and what you can see is in this case, when they've got they're entrained on a day dark cycle, they will tend to be pretty active as you're going from dusk into evening, and they'll be active throughout the evening, and then for most of the day, they're pretty much silent. We have other animals. Here's another animal over here. This is a little banner-tail kangaroo rat, which is cute. You can see he's got these cheeks that are jammed full of seeds and stuff. But this also, so this is a time frame. Each row would be a different day that they're measuring. But as they're moving from this side to this side of the graph, that's depicting the passage of one day. So here it's 12 noon. So this middle line would be midnight. Here would be noon again. And what you can see is that they're basically active at night. Humans, of course, are active in the opposite way. We tend to be most active during the day and less active during the night. So this is sort of the typical rhythm of a typical human. Not all of us are like that. I probably would be mostly nocturnal if I could be, but I can't be, so I'm not. So we're going to get back to those sort of concepts of circadian rhythms, and we're going to start with talking about sleep, so those times when we are inactive, because it's a really interesting neurological phenomenon, despite it being such a common part of our lives. But before we get there, I just want to talk about a common way that we actually measure these things. So, how do you measure activity of the brain in a person? I mean, you certainly can monitor their behavior, which is actually what those experiments that I just showed you are doing, is they're monitoring the behavior of an animal, and you can do the same thing with a person. You can monitor how much they move around, or how many words they say, or some other indication of their behavior that indicates something about their activity level and their alertness level. But we can also directly measure brain activity, at least in sort of a generalized sense, using a concept that's really, I mean, these are basically grandiose extracellular electrodes, and they're called electroencephalograms. So the idea is you take electrodes and you tape them all over your person's head, which is what they've done in this case. This is actually one of the researchers themselves, but a lot of times sleep researchers end up doing a lot of research on themselves because they're a convenient test subject. But he's got little electrodes taped all over his head, and he's snoozing away in, as your book puts it, in the comfort of sleep labs. So they have these labs that are dedicated to this. They try to make them really, really quiet and good situations, good places where people can sleep comfortable. And then they monitor sleep behavior. And as they're doing so, from these electrodes, by comparing the electrical activity that's sort of right beneath the scalp in the cranium, this mostly would be cerebral cortex activity, because that's the most proximal, closest bit of brain tissue that those electrodes would be near. You compare multiple electrodes, and you can get an idea of the differences in voltage, the activity that are occurring over time in that person's head. So in this case, they're showing electrodes comparing sort of the temporal sides to up here at the top. And as those comparisons are made, they run those signals through an amplifier so that they're detectable. And then here they've got a pen recorder, which is cute because that's how they used to do it. Usually now everything is digital, of course. But then they can monitor the little up and down signals. And each up and down signal corresponds to either a general increase in positive charges or negative charges just beneath where those electrodes are mounted. And it gives you sort of a summative measure of all the electrical activity within the brain, or at least in the parts of the brain that you're comparing. So again, here they're comparing sort of these two spots. In this trace, they're comparing these two spots. They might be different depending on what's going on the left side versus the right side of the person's head. So what exactly are EEGs measuring? Again, for the most part, you're looking at synaptic currents that are occurring right beneath the surface of the skull. And the largest synaptic currents that are made would be those synaptic currents that excite the dendrites of really large pyramidal cells. I'm going to say pyramidal because I can't say pyramidal. These are some of the largest cells. These include cells like Bett cells, for example, that we talked about in the motor chapter. But there's other sorts of pyramidal cells that are found all over the place. And these guys are, as they're present, these guys are receiving strong stimulation. And then these stimulations are causing changes in the excitation that is then detected up here by these electrodes that are placed on the surface of the scalp, or of the cranium. There's some interesting things about this. One thing they point out is that usually upward deflections, just by convention, you could do this either way, but since they've been measuring EEGs, upward deflections actually indicate more negative currents, whereas positive deflections are actually going down. So it's kind of the opposite of what my brain would think about. I'm not going to ask you to really interpret the up and downs per se, though. Really what we're interested in is what is the overall shape of the activity. Are we getting lots of small little short disturbances or big long wavy sort of disturbances? And that's oftentimes what we're interested in doing. I think it's really important to realize these EEGs are not picking up 1 or 10 or even just 100 of 100 different neurons at once. They're, each one is probably responding to the sort of summed electrical activity of literally thousands of depolarizations to get any kind of measurable effect. So you're not getting any good information about what any one neuron or even a small collection of neurons are doing. Really, you're doing, at best, you're comparing, what's going on the left side of the head versus the right side of the head, or the rostral versus the caudal part of the brain. But you're not getting really specific information for the most part about any one sort of smaller area of the brain. You can't measure what's going on in the hypothalamus using this method, for example. As I said, what we're often interested in is the general shape of these patterns, the EEG recordings that we're getting out of these measurements. And those patterns indicate something about the general activity of all the different neurons that are down in that area. So for example, you could get what look like lots of little irregular up and down sorts of motions, which is more like what this would be a recording that might be reflective of that. Or you could get sort of these big up and down sweeping sorts of depolarizations and repolarizations, apparently, kind of like this. So you can see, how different these two summed recordings are down here. And those recordings are basically a manifestation of all the different neurons that are present in that particular area. Here, and just to illustrate this, the book is showing 6 neurons in one place and showing how different kinds of activity patterns could result in different shapes of the EEGs. So in the top graph, this is showing what we would refer to as sort of more of an irregular rhythm. And what's happening here is that if you look at what each of these 6 different neurons are doing, so they got 6 different neurons, you could sort of imagine if we could record from each of them, which we could stab little intracellular electrodes in each one. Maybe this is what each individual neuron is doing. And I think the thing you can see is that each neuron is basically out of phase and doing different things than all the other neurons. They're not consistently all going up or down at the same time. So they're not experiencing the same sorts of stimulations and charge changes and membrane potential changes as the others. And so as a result, if you just sum across all these different ones, here you've got sort of a downward deflection, an upward deflection, a flat line, a downward, a flat, a downward. The summed effect is you get a bit of a downward spike here, but if we just move a little bit further along in the recording over time, you get an upward deflection of here, but other neurons will be going down. It's going, each segment they're going up and down, they're not doing the same thing. That's kind of the main point. And so as a result, you get this really, kind of chaotic, small scale disturbance sort of EEG reading. In contrast to that, sometimes that same brain area might be having much more regular synchronized types of action. So that's what's being shown down here, where you've got, we're seeing these large sweeping upward and downward deflections that seem to be pretty rhythmic and really they're manifesting from the fact that all the neurons that are in this area are more or less doing the same thing. So in this case, with this big, large sweeping upward deflection, the reason it's big and large and sweeping upward is that pretty much every neuron in that area is showing a bit of a positive deflection in their individual EEG, and then shortly afterwards it goes down because all of them go down. And so we have this sort of grand summed effect where when you see big, long sweeping patterns, it means pretty much all the neurons are in phase with each other, all doing the same thing collectively. So why might that be? I think what you're witnessing is basically different behaviors of these neurons. Oftentimes when neurons are specializing, they will tend to, they're doing specific discrete tasks in this area. So they're processing information. Each one is specializing in different kinds of interpretation in some ways. You'll get a situation where each one is responding in a different way. And so you get these really sort of chaotic, rapid up and down movements at the level of the whole EEG. In contrast, there are other times when they're all kind of playing in sync with each other. And in those cases, usually that means that they're not actually doing a whole lot of processing. They're all interconnected. They're all kind of responding in the same way. They're probably not specializing in any particular processing tasks, though. And so you get these kinds of actions. So these kinds of high, fast up and down motions tend to be more indicative of a really active brain or a really active area of a brain. So as you're taking a test and you're concentrating on something, your brain will tend to have lots of these little tiny fast up and down waves, which we'll name in a little bit. Whereas when you're sleeping, you tend to have these more long phasic up and down sorts of actions where all the neurons are kind of in phase with each other, but they're not really doing anything per se in terms of discrete processing. A lot of times what's happening in this kind of situation is you have some large, powerful central pattern generator that might be driving behavior in lots of downstream neurons at once. So all the neurons end up responding in the same way to each other. So your book makes an analog to this being a little bit like a symphony orchestra, where all the neurons are more or less in sync with each other. Even if they're not doing exactly the same thing, they're all responding to the same lead. And so you've got your conductor up there that's acting as the central pattern generator that's causing them all the neurons to behave in the same way. In sleep, one thing we'll see is that the thalamus can act as a really powerful central pattern generator. So the thalamus is like this central hub of sensory information coming into the cerebral cortex. So there's signals going from the thalamus to all different parts of the cerebral cortex, all the different sensory areas at least of the cerebral cortex, and even some motor areas. And so in this way, because the thalamus is so interconnected, if it starts to behave in a rhythmic fashion, it may cause whole parts of your cortex to start to behave in a rhythmic fashion. In contrast, other times you may have situations where there really is no discrete central pattern generator that's governing what's going on, at least in one area of the brain. Each neuron may be kind of responding to the neurons around them. Sometimes that can still lead to these sort of large synchronized up and downs if they're all interconnected and they're all basically, because they're interconnected, they're responding in the same way. But oftentimes you do get into situations where because each neuron's kind of doing its own thing, you'll get these sort of quick up and downs. Your book likens this to kind of like a jam session. So if you saw, oh my God, if you saw La La Land, did we see La La Land? It was so good. That movie just made me so happy. I don't know, even. It was just sort of an uplifting thing, and I think I needed that at that point in my life when I saw it. They talk about jam sessions with jazz bands, and those of you that are musically inclined know this far better than I. But in a jam session, everybody's not doing necessarily the same thing. Everybody's reacting to the other person and putting their own little interpretations as they go into whatever the song is that you're playing. And that's more or less what's happening here, where each neuron is interconnected and they're talking to each other, but they're all kind of putting in their own little interpretations and specializing in different ways to create the overall effect that we're measuring within EEG. So you can think of those sorts of scenarios as cases where you've got sort of the collective behavior, all the interacting neurons manifest in this kind of a rhythm. So I've been sort of hinting at this, but the cool thing about all this is that you can get a lot of information about general brain function by just measuring these EEGs. So as you change from different states, so the best example is you change from a waking to a resting or waking to a sleeping state, you will transition and see changes in your brain rhythms. Similarly, if you're just kind of hanging out, relaxing, not really thinking about anything, maybe you're daydreaming, versus then you start to concentrate on doing a math problem. In those situations, you'll also see changes in the brain EEGs, which is corresponding to different states. High frequency up and downs reflect specialization of neurons to specific tasks, whereas low frequency, highly synchronous, long sweeping sorts of EEGs indicate something about the interconnectedness of neurons, but probably doesn't indicate anything about major functions of them. Now, sometimes, it is normal for us to be alternating between these different states. You can run into situations where you have sort of extreme synchronicity between neurons across large areas of the brain. And in those circumstances, that can result in pathological types of conditions. So a good example of this is what happens during a seizure. So you can think of seizures as like a runaway excitation phenomenon where you have just large dramatic excitations and then inhibitions across large swaths of the cerebral cortex all at once. And so that means that all the various neurons are basically doing the same thing at once. That's usually going to result, I mean, in those cases, it's happening to such an extreme degree that it results in these sort of, it can result in these sort of chaotic movements that occur sometimes with seizures, or just the general locking up that occurs with seizures, depending on the type of epilepsy that we're talking about, or the type of seizure we're talking about. So these are recordings from an EEG from somebody who's undergoing a seizure. And you can see they're sort of starting with pretty normal up and down, fairly chaotic waves that indicate sort of normal daily alert function. And then they start to get these really large, huge, sweeping up and downs on their EEG across all the different sites across their head. So each of these numbers corresponds to different electrodes. And this is what you're seeing. And so during this period, they would basically be having a seizure where they're having, the whole brain is basically in sync with itself. It's losing function and, just sort of behaving in this erratic fashion at that time. Not all seizures are, fall on the floor, trembles. You can have seizures that are more sort of in the visual cortex, where you start to have, I think sometimes migraines might be related to this, where you have these sort of aura effects. I get very occasionally, it hasn't happened in about 5 years, but I used to get situations where half my visual cortex would apparently be undergoing some kind of a migraine. I'm not sure it's not this either, where like half of my visual field would just basically get kaleidoscopy. It's crazy. It was called a silent migraine with aura, and it's not fun to try to teach through. I've done it, but like write lectures or papers or something as I'm like, I really just need to lay down, but I can't always, don't feel like I can always do that. So not all seizures are, again, motor realizable in terms, or are realized in terms of dramatic motor convulsions, but they do seem to all have these characteristics in common, where there's sort of these large sweeping up and downs. There are many, many different causes of seizures. One interesting one is that generally speaking, GABA is an inhibitor that helps to prevent this kind of synchronicity, where GABA, we talked about GABA as a neurotransmitter, it's an inhibitory neurotransmitter, so it would tend to prevent strong excitation. And that's kind of what's happening, at least some of the time, is you're getting strong excitations across much of the entire brain. So if you have GABA inhibition, it helps to prevent that from occurring. So it's sometimes the case that people that have epilepsy, where you have sort of chronic seizures that are occurring frequently, can have either a lack of GABA inhibition from a problem with the receptors for GABA or the manufacture of GABA. Generally speaking, if there are drugs that function as GABA receptor inhibitors, and if you give a person that can sometimes lead to convulsions, because you're sort of mimicking this effect in the person where they don't have sufficient GABA inhibition that normally should be there to prevent these things. But tumors, mutations in ion channels, reuptake transporters, there's lots of different ways that you can disrupt something about the behavior of neurons across the brain in sweeping senses. And as a result, you can get these large synchronous up and downs that occur during and ultimately lead to epileptic seizures. All right, so any questions on EEGs so far? All right, so that's our tool. That's what we can use, these EEGs. We're going to use that as our tool to try to monitor the activity of the brain. And we're going to do so initially by looking at what goes on during sleep, because the EEG is such a helpful tool, because you can't ask a person what's going on in their head when they're asleep, because that's kind of the thing that happens during sleep, is you're sort of losing consciousness and you're not really aware of what's going on. So we can study what's going on in a person's brain using these EEG tools. I think sleep's really interesting. It's something like about a third of our lives. So if we live to be 80-something, a third of that time, if you count it all up, we're basically asleep. Or any more, maybe it's getting closer to a quarter as everybody gets themselves sleep deprived. But it's still just crazy that we spend that much of our lives asleep. And it's honestly just not completely clear why we're doing it, what's going on, what the purpose of it all is. Despite the fact that we spend enormous amounts of our lives doing it, and it's not just us, it's most animals, frankly, do this. Bless you. So for example, fruit flies, these little dinky fruit flies, Drosophila melanogaster, they sleep, as do most insects. They go through these sleep periods, and there's even genes that seem to govern these things. So this is a figure out of a different textbook, but I think it's really cool, where they're looking at this particular gene called the PER gene, which we'll come back to on the homework. This is one of these clock genes that seems to be important for determining their sleep cycles, and frankly our sleep cycles too. Here's the gene, and then here are 3 point mutations. And what this is showing is that a wild-type fly would be basically active during a discrete period of every 24 hours. So the wild-type normal fly that's not mutated has a normal sort of activity period and then a silent period each day. But you could have mutations where you have where they basically are active for longer each day. Or here's another mutation where they tend to be active for shorter periods each day. Here's another one that just has no rhythm at all. It's called an autorhythmic unit, or arrhythmic unit. And the thing is, these are individual mutations that all occur across this PER gene. So this PER gene seems to be really important in determining the sleep cycles of the fruit flies. And the cool thing is, fruit flies have that gene, but so do we. And it's pretty much the same gene. It seems to have very similar functions in us. It's involved as a clock gene. Dolphins sleep, and many marine animals sleep. Dolphins are kind of cool because the way they do it is they only sleep one hemisphere at a time. They never really like find a little nook and settle down and sleep. They're just sleeping in the middle of the water column. So they basically turn off one side of their brains at once. They'll even sometimes do a little bit of a roll so they can watch for predators down below. And they'll sleep on one side for an hour or two, and then they'll flip and sleep on the other side. And they kind of go back and forth for about 12 hours each day. And this is the way they do it. So animals are sleeping all the time. We sleep all the time. We have a tremendous amount of time. Sleep time for different kinds of animals can vary quite a bit. Some animals sleep more than 12 hours a day. Other animals sleep quite a bit less. Even people seem to vary quite a bit. So some people really do seem to just need about 5 hours. That's kind of on the extreme low end, but there are people that function just fine. Like if they didn't have a clock in their lives, that's what they would do, and it would be fine, and they'd function just fine that way. Most people need quite a bit more than that. Most people are needing somewhere in the vicinity of 7 to 9 hours. But there are some people that need even more. Really, some people to behave really effectively, they're sort of set up. So they really need like 10, 11, 12 hours of sleep to work really well. It's very difficult to get that in life, but there are people that that really is what their bodies seem to need. So there's that kind of variation. So I think sleep's really interesting because it is so variable, and yet it's so conserved. We see so many different people and different animals that are all participating in it. And yet it's kind of unclear why we're doing it, what's going on in our minds as we're doing it. There's all kinds of mythos around dream interpretation and stuff that occurs. I mean, dreams in general are just weird. Like why do we dream? That's a really interesting question. So the goal here is going to be to try to dive into this question about sleep and try to understand, at least neurologically, what's going on. I'm not going to be able to explain why you dream, but although we have some hypotheses. I certainly am not going to explain whether or not there's any meaning to them. And I don't want to get into discussion of dreams too much in here. But that's kind of where we're going this week. So I'll see you guys Wednesday. I think it's a good setup for that.